Hello, everyone, and welcome to today's talk titled Timing is Everything, Biomarkers for Early Detection of Preclinical Toxicology Events, presented by Sapiens. The ability to detect biomarkers of adverse drug changes early in preclinical studies can support better and faster go, no-go decision-making. This presentation will explore how small molecule biomarkers can be leveraged to detect drug-induced toxic events well before the histopathological changes are observed. To kick things off, I'd like to introduce our speaker, Dr. Phil Warboys, Head of Translational Science at Sapiens. Dr. Warboys is a drug discovery and development professional with over 25 years of experience in pharmaceutical research and development, including deep expertise in research and translational science. I know I'm looking forward to hearing his insights today. So with that, Dr. Warboys, please take it away.
Yeah, thank you very much, and it's a pleasure to be able to give this talk. Really appreciate it. So we'll start with this slide, which is really focused on the future of safety assessments and the role biomarkers can play in this. So I won't belabor the point, starting at the top left about the increasing drug development costs. I think everybody in the industry knows this, and toxicology studies are an important part of that cost, not just because of the actual dollars that are spent, but the timeline costs of failed drugs and then backup molecules coming forward, and what that means to the overall drug development timeline with the billions of dollars that are involved. There's also the aspect of 3R. We're all trying to reduce the number of animals, refine and replace where we can. And a couple of years ago, during 2020, there was a survey conducted by the IQ consortia, DrewSafe in particular, which was published in 2021. And this on the right side highlighted the desired future state with biomarkers. And you can read many of these bullet points, but there was some in particular that I think are important as we Sapient consider, our role in this. And in theory, we can impact all of these, but I think the ones here highlighted are, more of the near-term areas that, we as a company are focused on with the technologies that we bring to the industry. And in the coming slides, I'll cover how we think the application of small molecule biomarkers to some of these areas, including speed to market and decreased drug attrition, can be made through the increased use of biomarkers in preclinical safety assessment. I think an important aspect of the small molecule biomarkers is the dynamic nature. So these rapidly change in the in vivo setting, and they respond rapidly to changes due to toxicity that can be induced by a drug long before it may manifest in terms of histopathological changes or some of the downstream biomarkers that are already detected and monitored via clinical chemistry readouts. And this ultimately drives the goal to look early for markers of toxicity, so drug development programs can be optimized to pick the winners and obviously remove the less favorable drugs from the pipeline. So why specifically small molecule biomarkers? There's several reasons why we believe they lend themselves to the assessment of toxicology, particularly in preclinical species. These molecules by definition are small and they're readily released from tissue into blood, either passive permeability or via transporters depending on the organ. So they're continually moving in and out of the bloodstream and tissues and represent a readily accessible matrix in the blood that you can sample from them. They also reflect changes in the organs. So depending on whether it's a disease, if in the case of human, or toxicity in the case of animal, different molecules can be released, or the same molecules can be released, but at just different levels, either increased or decreased. Another important aspect of these small molecules, which really represent a lot of our internal biochemistry, is that they're conserved from cellular systems through the preclinical species to humans. And this table on the bottom left, captures some of the chemical classes that are measured, and they reflect many of the core biochemical pathways that occur, in cells, in animals, in human, to differing levels, but they facilitate the translation and the human relevance of toxicology findings, potentially to human. So this slide really captures something that I think is fundamental to why the approach we take, is exquisitely suited for the study of toxicology, particularly, animal studies of toxicology. So on the left is the human situation. So the approaches that we take to discover biomarkers of human disease and drug effect in human are well established, they're well documented, and they really have been used across many therapeutic areas as part of drug development. I think a key thing here is we're able to make those discoveries despite a very heterogeneous background. So in the human population, the genetic variability obviously is fairly high, but particularly high is the environmental variability. So this can be diet, geography, age, BMI, previous therapeutics, etc., etc. There's also variability in terms of sample collection. So how a sample is collected from one person last year versus another yesterday, there's variability. Yet despite all of this variability, we're able to discover very powerful and very specific biomarkers in the human population. Contrasting this to the right in animal studies, much of this variability is dramatically reduced. Preclinical species, whether it's a rat, a dog, a mouse, a monkey, they're inbred strains. So the genetic variability is obviously, drastically reduced, basically zero relative to human. Same with environmental variability. Animals are housed in the same room, they're fed the same diet, the same, age, etc. So the environmental variability is drastically reduced relative to human. And also the sample collection variability. They're in laboratory conditions, very tight control over how samples are collected and processed. All of these factors reduce the background heterogeneity to give a very, very low noise threshold, so to speak, to be able to detect changes that are a result of the dosing of a drug in a toxicology study setting. So how do we actually go about discovering biomarkers? So this is the actual lab that Sapient has in San Diego, California, and it's based on these mass spec LC systems. So on the front end, we have what we call our rapid liquid chromatography system, coupled to these very high-end mass spectrometers. So these are Bruker timsTOF Pro 2 machines. They allow us very quickly to capture a very, very broad range of chemistries, as was documented a couple of slides back in that table. So we capture molecules from very polar to polar lipids to non-polar lipids to capture, in effect, the broadest range of biochemistry possible. And within a sample, we typically get 11 to 15,000 individual molecules from, for example, a plasma sample. So some of those molecules are already known, so they're standards that exist, and we can annotate those standards. The vast majority of molecules that are circulating in somebody's blood or in an animal's blood, though, are actually unknown. They're relatively low levels, they're very potent molecules, and historically, because the sensitivity of the equipment isn't what it is today, they just haven't been picked up. But with the advent of equipment like this, we're able to detect very, very low levels of extremely potent molecules to make new discoveries. And these are what we call the unknown molecules. And these allow us to track many, many processes that are occurring within cells and within organs to facilitate then, the discovery of biomarkers related to, in this case, a toxicological event. Okay, so I'll give an example of how we've used that in a very focused way to address a toxicity finding within a particular species in the context of lead and backup molecules. So if we consider this example of a lead molecule hypothetical structure with a couple of backup molecules, a typical drug discovery development program begins with, maybe a couple of week toxicology study where, a candidate drug is nominated. Obviously, no major findings, the drug progresses into potentially a 4-week study and ultimately into maybe a 13-week study. Obviously, these times can change, but this is a fairly standard paradigm. It takes several years because this is interlaid with a CMC program, the clinical program, etc. And sometimes what you get at the end of a 13-week study is a no-go. So a new toxicity could arise, or margins suddenly collapse relative to the 2-week and the 4-week study, which were looking good. So this obviously throws a curveball into, a development program, and is one of the things behind the increase in drug costs in terms of the time and the money invested to get to this point in drug development. So the question is what to do about it. So clearly a program has backup molecules. The question is, will those backup molecules suffer the same fate? So by definition, they have the same primary pharmacology as the lead molecule. They're designed to hit the same target, and they come from sometimes similar or the same chemical classes. Medicinal chemistry groups, assuming we're talking small molecules, try to design in structural diversity, but because you're going for the same target, there's only so much diversity can be built in. So the question is, will they suffer the same fate, and will we only see that fate in a late stage study? This is where biomarkers, can be extremely important to try and get an early readout on backup molecules to see whether they de-risk the issue that was inherent in the lead molecule, or they carry the same fate. The goal here is obviously to, make a decision faster, reduce the investment that is needed to get to that potentially kill outcome, and speed programs along by killing molecules early and picking the winners. So the way we do this is actually utilizing the studies in the lead molecule that have already been done. So there's a very, very rich source of samples from, in this case, as an example, the 2-week study, the 4-week study, and the 13-week study. Toxicokinetic samples are typically taken at the beginning and the end of those studies. So we have longitudinal samples at multiple dose levels from which to make discovery. This is a very, very rich source of samples, particularly given the low genetic, environmental, and sample collection variability relative to human. So we would take all of these samples, let the animation go, okay, and we do our non-targeted discovery RLCMS. So we capture all of the molecules that our systems are able to pick up from all of those samples, and we combine them with the safety data tables. So the results of the toxicology studies, the 2, the 4, and the 13-week. Biocomputationally, we integrate all of this information into something like the volcano plot in the lower middle part of this slide. So this looks at the change relative to dosing and the statistical significance of all the molecules that are measured. And as you can see on the right, some molecules are highlighted in red and blue. From the mass spectrometers, we're able to look to see whether these molecules are chemically interrelated, some of them. If they are, they can be indicative of various points on a biochemical pathway. But ultimately, we want to narrow down to a biomarker or a couple of biomarkers that are indicative of the safety finding. And here we would focus on the top right, the little red biomarker that's circled. And at Sapient, we would take that information on that molecule and move it to develop a targeted assay for that molecule that can then be used in the drug development process for the backup molecules. Now, the way we get to that is always a question. People say, well, if you've got an unknown, how do you get a targeted assay? So this slide considers the situation of the known and the unknown. So, within our mass spectrometer, we pick up about 1,000 known molecules. So these are standards for which we've run them down the system, and we can very quickly annotate those molecules and say, with standard that has been purchased, we can very quickly move to a simple triple quad type of assay, akin to measuring the drug analyte itself. In the case of an unknown, though, which are by far the majority of the molecules, a little bit extra work has to be done. So firstly, we would do some detailed fragmentation of the molecule to start to build knowledge of the structure, ultimately leading to isolation of the compound. So we have PrEP LC systems where we can isolate particular compounds at scale and then take them to NMR spectroscopy and ultimately synthesize a standard. This standard can then be used to develop, again, a targeted, validated assay, whether that be a non-GLP or a GLP assay, depending on the application. So once this biomarker is identified, it can then be used for the backup molecules. So in this case, backup molecule 1 showed a change in the biomarker that was detected early in the samples that came from the lead molecule, indicating the potential that this molecule may suffer the same fate as the lead molecule. In the case of a different backup molecule, no change in this biomarker was detected, and the program would proceed into the clinic and into longer-term 4 or 13-week toxicology studies, with the goal that this would have a clean readout relative to the lead molecule because of the absence of a change in the biomarker early on that was discovered from the lead molecule samples. Another key question that can be addressed with biomarkers is the human relevance of a toxicology finding, particularly if it occurs in a single species. So sometimes your finding occurs in rat, but not in dog, or vice versa. The question is, which one is most human relevant? Here again, biomarkers can start to build a weight of evidence approach for, is a finding specific to a particular toxicology species, or is it representative of the clinic? So here we would again take the samples that were profiled, in this case from the lead molecule, and here with the liver finding, we have 3 hypothetical biomarkers on the left, which are all increased in the situation of a toxic insult to the liver. Sapien has a very, very large human biology database of 6-figure sum of people for which we have all of their medical information as well as all of their metabolomics information. This covers a wide range of human disease. Here I've just focused on liver. So within our database, we have people who have various insults to their liver as measured either clinically or via clinical chemistry. And we can ask the question, relative to the changes in the toxicology species, how do those same biomarkers map into our human database? Are those same biomarkers present? Do they increase, decrease? The other thing the database allows us to do is to strip out other effects that are present in humans, diurnal rhythms, confounders such as renal dysfunction, it could be cardiac dysfunction, age, BMI, etc. Our database is extremely powerful to ask the question of how does a preclinical finding map into a human population? So another thing it allows us to do as well is conduct risk assessment in humans. So once we have identified that biomarker preclinically and developed a targeted assay, we can use it to monitor in human samples, particularly the first in human samples where dose escalation often pushes to relatively high levels to gain an understanding of how that risk that was observed is or is not translating into humans. Once those samples are collected from a phase 1 study, they can also be profiled on our rapid LC-MS systems. This chromatogram on the right gives you an idea of the actual output that we get from these systems. So again, we get the polar metabolites, polar lipids, and non-polar lipids. So we get thousands and thousands of molecules that give us an insight into how a test drug is behaving in those initial healthy volunteer subjects and allows us to triangulate back to the preclinical tox species. One key question is fundamental to drug development is the toxicity we're seeing on target or off target? If it's on target and it's exaggerated pharmacology, And chances are the backup molecules, because they're targeting the same mechanism, will probably have the same finding, and it becomes then a question of margin. If it's an off-target mechanism, though, then medicinal chemists can try to design out certain off-target potencies relative to the desired target. So being able to tease this out, can be a real key to deciding on whether backup molecules, should be reworked or progressed forward. So again, here what we would do is take the findings from the GLP toxicology samples, where some biomarkers are increased or decreased because of the dosing of the drug, and compare them to other samples, either phase 1 samples, other preclinical PD samples from pharmacology models, or cell models. And here we would look for which biomarkers are changed by the test drug, and do they overlap, or are they different? Do they directionally increase or decrease? And by combining both toxicology samples with preclinical and clinical PD type samples, you can start to build an understanding of whether the toxicity you're seeing in your preclinical tox species is most likely to be related to on-target exaggerated pharmacology outcomes, or is actually an off-target binding that is occurring, and then whether that can be mitigated via changing the structure of backup molecules or not. So I'll wrap up with this, which really says that the tool we have can be used in multiple ways. So it can be addressing late stage findings. It can be used to assess the translatability, understand whether you're looking at exaggerated pharmacology or off-target effects. But also it may lead to early dose adjustments. So sometimes in a toxicology study, signs of distress can be evident in animals, and the decision, is whether to, drop the dose or terminate the high dose group, for example. The use of biomarkers early on can give an early idea of whether animals are heading down that path based on findings that have been seen with, say, a lead molecule, and then decisions can be taken, whether to dose reduce an animal or take down a top group. So there's multiple ways of applying this technology and bringing biomarkers into toxicology studies to facilitate the drug development process, and ultimately, of course, adhering to the 3R initiative. So, we can refine, replace, and reduce the number of animals that are used in toxicology studies. So with that, I will say thank you and pause for any questions.
Thank you so much, Dr. Warboys, for that wonderful presentation. We do have a couple of questions, so let's get started with that. The first question for you, your examples focus on plasma analysis. Can you also analyze tissue samples, and are there any species limitations?
Yeah, absolutely. So those examples did focus on plasma, and plasma definitely makes up the vast majority of our human biology database, but the equipment is amenable to any sample that can be got into a liquid form. So historically, we've analyzed CSF, tears, aqueous humor, urine, but also tissues. So homogenizing liver, for example, or stool samples. The only difference with those sort of samples is there's just a homogenization and an extraction step before the extracted sample goes down the mass spectrometers. Likewise with species differences, as far as the mass spec is concerned, they're all the same. So a plasma sample from a rat or a dog or a human is analyzed exactly the same way. The only difference is obviously the biocomputational aspects on the back end. So the tools are totally amenable to different matrices and species.
Great, thank you. Next question, what sample volumes do you typically need?
Yeah, so the whole process from sample extraction through to the mass specs is totally automated. So if we consider plasma, typically 150 microliters is what we ask for. The amount of sample that actually goes down the mass spectrometer is actually well under a microliter. That volume of 150 microliters just facilitates the automated sample extraction. But where sample volumes are limited, CSF, for example, then we just manually pipette those. So anything down to 10, 20 microliters, we can handle.
Thank you. The translation example you presented was specific to liver disease. What about your ability to support other disease areas?
Sorry, could you repeat the question? I think you broke up slightly.
Absolutely. The translation example you presented was specific to liver disease. What about your ability to support other disease areas?
Oh, of course, yes. Yeah, so again, go back to, if it's a particular organ that is of interest, kidney, liver, heart, for example, because we can homogenize and extract those tissues, then, we can look at other organs that could be affected in terms of a toxicological process. Likewise, translate into our human database, we have people who have got various types of kidney disease, for example, or muscular issues. So we are, I would say, therapeutically area, or in this case, disease or toxicology agnostic. Our equipment, can look at all aspects, and our database captures a broad swathe of the human population in terms of facilitating translation from different organ toxicities into human relevance, so the clinic.
Great. I think we have time for one more question. So last question, there is a lot of interest in technologies to investigate mechanisms. How do you see other tools overlapping with discovery metabolomics?
Yeah, very good question, because ultimately the question is, what is behind the toxicity that is being seen and the human relevance of it? So this gets back to one of the points I made earlier around these small molecule biomarkers in terms of their conserved nature. So between cellular systems through to in vivo animals through to the human situation, we're measuring in many cases similar biochemical pathways and looking for perturbations within those pathways. So when you go to try and understand and tease out the mechanism of a toxicity, often it's bringing in orthogonal experimental systems. So it could be cellular assays, it could be things like organ on a chip experiments, as well as in vivo experiments. These small molecule biomarkers allow the linkage of those different experimental systems that are asking different questions. The combination of them all and looking at the conserved biomarkers across them can often allow you, through a weight of evidence approach, to actually tease out what the mechanism behind a toxicity actually is. So I think it's essential to get to the answer of what is causing toxicity, that you bring in other experimental conditions. It's very rare that one experiment alone will give you the total answer. It's the accumulation of human information, cellular systems, and other ex vivo models, as well as in vivo models.
Thank you so much. We just want to thank you, Dr. Phil Warboys and Sapient, for joining us today with this wonderful presentation. For all of our attendees, this has been recorded and will be available on the AAPS website. And thank you again, and we look forward to seeing you all at a future eChalk Talk. Thanks so much. Have a great day.
Thank you.